why should you be interested in genetics? Well, there are all these questions posed by genetics that you can sort of answer with them. And frankly, the way we're going to be looking at genetics, the way this textbook looks at genetics, is slightly dated. Some of these techniques and things are a little bit old, but they're still foundational for the way that we understand things. They're still very useful pieces of knowledge to have. So some of the questions that get posed by genetics are, how can sickle cell anemia be both deleterious and protective? Um, how should a woman's, or why should a woman's chromosomal makeup protect her against color blindness? I don't know if you guys know any colorblind people, but it is rare for men to be colorblind, and it is extremely rare for women to be colorblind. How can a healthy parent give their offspring an extra copy of a chromosome, which is what happens in Down syndrome? There are other conditions that involve that as well. And it is estimated that we have 5 to 10 genes with mutant alleles that cause disease. Why are genetic disorders relatively rare? So to kind of start talking about genetics, we need to start here with Thomas Hunt Morgan. So we've talked about guys like Gregor Mendel, who discovered that there were these maybe genetic components to what makes us us. And he thought about these ideas of there were maybe these alleles. But Thomas Hunt Morgan was the first person to find that genes were transmitted differently in males and females because some genes are located on the X chromosome, which we're going to talk about later. And he studied the frequency of recombination and figured out how to determine the distance between two genes based on how frequently they recombine with one another. And we're going to talk more about this later on. We're going to talk about this idea of recombination and what it has to do with crossing over. So Thomas Hunt Morgan studied fruit flies, and most fruit flies have these characteristic red eyes. But what he did is he basically waited for a white-eyed fruit fly to show up. So you have this white-eyed fruit fly here. And this is a result of a mutation on the fruit fly itself. So you see here you have a male, which is YX, and you have a female, which is XX. Those are your chromosomes determining what sex they are, right? So males are YX, females are XX, and he crossed these two with one another. He crossed these two, and this is a male that has the white eye phenotype. Cross them, and then you can see from this this Punnett square, some of these organisms are going to wind up with white eye alleles. They're going to wind up with one white eye chromosome or one white eye allele there. And you can see here the males that result from this cross. What color eyes do the males have here? So let's think about it like this. This is a recessive gene. This is a recessive trait. So in order to actually have the trait, or I guess I could say this trait demonstrates complete dominance. So in order to have the trait, you need to have two recessive alleles. So the males have just this dominant allele, and then they have this puny little X, or this puny little Y chromosome. This puny little Y chromosome here. All right, so the result of this first cross between the white-eyed male and the red-eyed female, you don't wind up with any white-eyed individuals at the end. And Morgan thought, well, that's kind of a strange thing. So what he did was, he said that it doesn't follow standard Mendelian genetics. And what he did was he took those individuals from that generation and crossed them back with one another. He crossed them back with one another, and what he wound up with is 50% of males are winding up with white eyes now. Keeps blocking it, that little thing. But 50% of males are winding up with white eyes. 50% of males are winding up with red eyes. And the reason for this is because the white eye gene is on the X chromosome. So remember, males are YX. And as we'll see, and hopefully I'll get a chance to explain in lab, the Y chromosome in males is puny. It doesn't have as many genes as the X chromosome on it. And so if you wind up with a male that has one of these chromosomes, one of these homologous pairs that has a white eye gene on it, the male winds up having white eyes because it doesn't have the red eye chromosome to compensate for it. Right? It just winds up with white eyes. That's all it has. It's all it can get. It's white eyes. This male over here wound up with red eyes. It's one of these, 50%, because it has this red eye chromosome, and then it has a Y chromosome with nothing there. There's nothing to compensate or change for it. These males wind up with red eyes. I'm sorry, these are females. These females wind up with red eyes because they wind up with one dominant allele. This has two dominant alleles. So X-linked inheritance is what we're talking about. This is a phenomenon that Morgan kind of discovered. There's a few examples of this. Hemophilia is caused by this mutation in a gene that codes for blood clotting factor protein. Duchenne muscular dystrophy is caused by a mutation on the X chromosome. Red-green colorblindness, like I was saying earlier, you meet some men that are colorblind. You do not meet many women at all who are colorblind. So those red and green pigments are made by proteins on the X chromosome itself. So mutation in genes for these pigments result in the inability to see color. These mutations are recessive because you only need one good copy of the gene for color vision. Okay, this is a red-green colorblind test. If you can't see that there is an 8 right here, you are probably colorblind or have some form of this colorblindness. So again, these affect men much more frequently than they affect women. Like I was trying to explain before, women have two X chromosomes. So the homologous pair in men is an X and a Y. The homologous pair in women is X and X. So if you have a dominant trait and you just have one copy of the X that has it and you have one copy of the X that doesn't have it, you're normal. You have normal vision, okay? Because your other X is able to compensate. Men, if have one mutation or they have a mutant X chromosome, they wind up with this condition. They wind up with the trait because they cannot compensate for that other X chromosome because they have this puny little Y as the homologous pair. So again, here is color blindness. Here is a mother who is not colorblind, but she has this colorblind mutation on her X chromosome. So the reason she's not colorblind is over here is because she has this functioning allele that gives her normal color vision. Then dad over here has normal color vision. He's not colorblind at all. He's got his normal X chromosome. He's got his puny little Y down here. And so let's look at the offspring they produce in this Punnett square. So how many of the daughters here are colorblind? None of them are colorblind because they have one X that is normal vision and one X that is abnormal. But again, it's like if you have big A, little a, what phenotype are you going to express? You're going to express the phenotype for big A. You're going to be fine. Whereas down here, the boys, this boy is fine because he has this normal color seeing allele from his mom. And in this one, he's colorblind because he has this X, and then he has a Y chromosome that can't do anything. It doesn't have the same alleles on it. It doesn't have the same genes on it that you see normally in a homologous pair. And so 50% of the sons wind up being colorblind, whereas 0% of the daughters in this cross wind up being colorblind. In order to have, sorry about that. In order to have a daughter that's colorblind, you need to have a mother who is a carrier, and you need to have a father who is colorblind, and that's a much, much more rare thing to happen. So in a minute, I'm going to talk about autosomal genetic disorders. These were X-linked genetic disorders we were just talking about.